In the last lecture, we already studied about the genotype, phenotype, and all the things. Now we are going to study about the pedigree analysis. So what is the pedigree analysis? Pedigree analysis is nothing. What is the pedigree analysis? Pedigree analysis is nothing. The analysis of trait in several generation. What is the any particular trait or character we are seeing in the several generation, which is being in the particular family, which is represented by certain diagrams. So diagrammatic representation of particular certain characters or certain traits of several generation of any human family, to just see the particular inheritance of character, trait, abnormality or disease particular order is called as pedigree analysis. So what is the pedigree analysis? Here the further symbols are there to show the pedigree analysis. Here you all could see the particular square is for male, female for this particular circle. Then sex unspecified means either male or female. It is not just specified. Then the particular sex unspecified character is being there. Then affected individuals, if the particular any disease or any particular thing which is being affected, which is being shown in the individuals, then it will be with the dark this color. Then the mating will be shown with this particular color. Then heterozygous male means with the character or just having. Heterozygous female will be circle. Mating between relatives, if there is relatives between mating occurring, then this particular diagram is being shown. Parents above and children below in order of the birth left to right, then this is parents with the most child infected with the disease, any particular disease being particular spread to the child, then it will be shown in the bold letter. Dizygous twins and monozygous. Dizygous twins means both are the twins but they are having morphologically different structure and shape. Monozygous means both are the twins which having same, means both are the girls. Dizygous means either one boys and girls but both are twins, but here both are girls. 5 unaffected offspring will be presented by this. So these are particular few diagrammatic representations. The particular diagrammatic representation is being given here, which is showing the pedigree analysis. So this was the pedigree analysis. So this you all had to study was about the pedigree analysis. What is the pedigree analysis? Now coming to your topic, coming to your particular topic, we are just coming. Pedigree analysis is the diagrammatic representation of a family chart showing the inheritance. What is the pedigree analysis? Pedigree analysis is nothing, just diagrammatic representation of family chart showing the inheritance. So these all are the particular examples of the pedigree analysis. So now Punnett square we already had studied that it is given by R.C. Punnett, where we just put one side the female, one side the male, and just we cross the particular thing so that that give the particular cross, make cross easier. Now coming to the sex linked inheritance. What we understand by the sex linked inheritance? Sex linked inheritance just means, or it just signifies, sex linked inheritance just signifies that disorder due to the heredity. Any particular disorder which is occurring due to the heredity means transferred from generation to generation. It is just transferred through the recessive gene which is present on the X chromosome. Any recessive gene which is present on the X chromosome, or the particular disease, genetic disease which is present on the X chromosome. We all know that X chromosome and Y chromosome are the two chromosomes. XX is present in female and XY is present in the male. But X is the criteria which is present in both male and female. So that particular, any particular gene which is particular present on the X chromosome, that is called as the recessive gene. So there are the particular few type of disorders, few type of genetic disorder which we just here discussed here: hemophilia and color blindness. Hemophilia is a particular disease, here the genetic disorder where the sufferer means if the female is homozygous, recessive female, and the recessive X bearing male, so there is the continuous bleeding. The bleeding is not stopped because the clotting factor is not present there. So that particular clotting, due to the absence of clotting factor, the bleeding is just started, is a genetic disorder, even though death also occur in the particular cases. And the color blindness is a case where the particular affected individual can't recognize between the red and green color. So they will not recognize that what is the red color and what is the green color. So 3 color circle in the chapter sense organ which were given, the particular they will not understand the particular, the traffic symbol they will just confused. So that is a color blind genetic disorder. Now here the squares is being given. Here you all could see there is a normal female where the XX chromosome, and color blind male. So what is happening? One side in the Punnett square we are keeping the male individual, color blind male individual and one side XX. When we are crossing, then we are saying when the female was born, the which is having only X1 carrier, one on the one X only, it is just carrying the C, means the color blind character. So that means that it will be not affected, it will be carrier for to transfer to the next generation, but the male will be normal. Why? Because here male, the X was combining from the mother, the X was combining, and father Y was being taken. So that is why the normal male will be seen. So now we were studying about one, we have seen the normal male and the other cross where the color blind female is there and the normal male was there. So what is happening when the color blind female is there? Carrier daughter will be formed, but when the mother is color blind, then X will be coming from mother only. So the son will be color blind, son will be occurring when the it will combining with the carrier female. Here the two crosses is being given. See, normal mother and the color blind father. Then when father is color blind, then the daughter is carrier, and when the mother is color blind, then the son is just color blind. And in one cases where the normal X, which is the here 2X is present, so when there is the normal X present, then in the normal X the child is not affected by the color blind. So this is the particular case and particular possibility of the particular thing. Here one chart is also given for to draw the possibility. Here also when the X0 and X will be, you will just type XX and then X0. Then what will be happening? When the XX will combine, then one the color blind female will be there, one the carrier will be there, then one will be the color blind male and one will be the normal male. So this is the particular characteristics or the crosses which we are just observing by the Punnett square. So this is the particular genetic disorder. Genetic sex-linked inheritance means the appear of the trait which is due to the presence of an allele either on the X chromosome or on the Y chromosome. When any particular allele is being present on the X or Y chromosome which the transmit or show the character, then it is called as a sex-linked inheritance. So after this we are just coming to the Mendel experiment on inheritance. We all know that inheritance means the particular character which is being transformed from generation to generation or inherited character. So here Mendel, we all know that Mendel has taken the Pisum sativum. Mendel was Austrian monk. He was who had just given his full times to the P, and he was just wanted to become a teacher, but he had become the father of genetics. Who was knowing that he will become the father of genetics? So Georg Johann Mendel, in the mid 19th century, was an Austrian monk. He conducted breeding experiment on the garden pea, which is known as the Pisum sativum, and his finding was the milestone in the biology, due to which we are studying today the genetics. So Mendel has selected garden pea. Why the Mendel has selected garden pea? Here the particular reason for that particular selection. Why he had selected on the garden pea? Because many varieties were available, means alternative form. If the just we are choosing the t-shirt na, so if the particular design t-shirt we are getting the red t-shirt also, blue t-shirt also, black t-shirt also. So similarly for the alternative traits he was having many varieties. Varieties were available in the true pure form. The true breed was present. Peas were normally self-pollinated. Pea was having the quality that it could be self-pollinated also and cross-pollinated also, and it has very short span of time, so that in a particular year we can rope more than one type of one time we could rope the pea and will study this particular thing. So Mendel took varieties of pea and he has shown the monohybrid cross and dihybrid cross. He had taken the 7 character. Here the 7 character is being shown here. Flower color, here see dominant recessive, means one will be the dominant and other will be recessive. Seed color, seed shape, pod color, then pod shape, flower position and plant height. These are all the particular character which Mendel has taken. Now first of all we are going to see about the monohybrid cross. Monohybrid cross means the particular one character will be crossed with the other character. What is happening here in the monohybrid cross? We all could see what is happening in Mendel experiment. When TT means a tall plant is being, when the TT is tall plant is being crossed by the dwarf, tall, and this will be dwarf, then in the F1 generation all the plant will appear tall. All the plant will appear tall means the dominant character will express itself and recessive character will be just masked up by the dominant character. So this will be the F1 generation. And when we are coming to the F2 generation, means when we are crossing TT with the TT. So what we are saying, when we are crossing this TT with the TT, then we are getting one dominant T, means both the T are dominant, one T and one T recessive, one T recessive. But here also phenotypically 3 will be shown tall because the capital T is being present and 1 will be the dwarf. So what will be the ratio? 3:1. Morphologically, phenotype means morphologically character. So that will be the 3:1, means 3 will be tall and 1 will be dwarf. But genotype, when we will study its genetic character, then in the monohybrid cross we will get 1:2:1, means 1 will be the tall, 2 will be the hybrid, and 1 will be the dwarf. So this is the particular genotype and phenotype of the monohybrid cross. Monohybrid cross, why? Because one character we had taken one time and we had just crossed it, and then after the F1 generation we had got all the tall plant, we had self crossed that plant, and in the F2 generation we had got this particular ratio. So this is the monohybrid cross. Coming to the dihybrid cross. In the dihybrid cross we had taken two type of character. Here round yellow seed means RRYY, and wrinkled green seed is being taken. For the capital R, wrinkled is being shown with the small r, and for the yellow dominant character, the green character is being shown by the small y. What is happening here? After the crossing, we are just getting in the F1 generation R small r, y, y. After the self crossing, we are getting four character: R, y, capital R, capital Y, capital R, small y, capital R, small y. Then we are getting small r, capital Y, and small r, small y. Both the character we will getting both the side, and after the cross we are seeing that particular cross. What the ratio we obtain? 9:3:3:1, means round yellow. If one capital R is present in any character, then that will be the round, because the particular capital R will express itself. So here this is the round, this is round. What is happening? If you all want to see, then you all could see that particular. If we are just seeing the round character, let us see the round character. Round yellow, we are just seeing the round yellow particular. So what is this? This is the round yellow, because here capital R and capital Y. Mark it, capital R, capital Y will be the round. Capital R, capital Y will be the round. Now here capital R, capital Y. No, this is not this particular capital R and capital Y. See here, capital R, capital Y. Then round yellow, round yellow, round yellow. Any more? Small r, small y, no. So 1 is 2, 3, 4, 5. Okay. Here also 1, here also 1, here also 1, here also 1. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So what we are getting? 9 round yellow. We are just seeing the 9 round yellow. Then now coming to the round green, means round means capital R and small y. So this capital R, small y, round green, it will be the round green. So we will just putting the round green for the square bracket. Round green, okay? Round green. So this is the three character of the round green. Then wrinkled green means small r and capital Y. So small r, capital Y we are getting, small r, capital Y, small r, capital Y. So just we are getting the particular round green, and one will getting wrinkled green. So in the dihybrid cross, what we are getting? 9:3:3:1 ratio. Phenotypically, its genotypic ratio is very broad, so we are not studying its genotypic ratio in your syllabus. So now there are the three laws. After these two crosses, Mendel has postulated three laws. The Mendel three laws were the law of dominance. With these crosses only he had given this particular law. Law of dominance was there, law of segregation was there, and law of independent assortment was there. Here see the particular cross which I was just showing, it is being depicted here. The round yellow seed and the wrinkled green seed. Both type of character in the particular dihybrid cross. You all could see or refer to your book. Okay. Now Mendel law of inheritance. Mendel law of inheritance is being categorized into three laws. Mendel law of inheritance is being categorized into three laws. The first is the law of dominance. Law of dominance was the Mendel was just not using the word gene. He was using the word factor for the gene. So he had said when the two particular factor is being combining, then the one which is expressing itself is called as dominant, and when which is gets suppressed is called as recessive, which is being shown here. This is being expressing itself. This was expressing itself. This was just expressing itself. So that is why this is particular capital T is dominant and small is recessive. So this is the law of dominance. Here dominancy is just expressing itself. Now law of segregation, what is telling? Then when two member of pair of factor means the particular allele or gene, particular factor which was used at the term of Mendel, particular factor separate during the formation of gamete. What he was saying, that particular at the formation of gamete they will get separated, they do not belt but segregate or separate in different gametes. So what is being formed? When here we are just combining in the F2 generation, what is happening? They are just segregating, separating, and each and every character is being shown, means the dwarf character is also shown. So this is the type of segregation, means the law of purity of gamete. When they are just self-cross, then they are just segregating its particular characteristics and just independently it is showing its particular character. Means dwarf character which is masked, here it is showing its particular character. Okay, now the third law is law of independent assortment. What is happening in the law of independent assortment? When there are two pair of character, the distribution of allele of one character into the gamete is independent of the distribution of the allele of the other character. Means here when it is just showing the dihybrid cross. What is happening? We are taking here in the dihybrid cross, we are taking here the two characters, round and yellow. Means round the shape of seed and the yellow is the color of the seed. So both character is not intermixing, means not blending. Both character have the capacity to express their self, means round and green or wrinkled and green, each and every character is being shown. So independent assortment means what? It have the capacity to show its independency, to show its particular character. So this is the particular type of three law. The first law was of the law of dominance. Law of dominance was there. Then the second law of the law of segregation, where the gamete is being without any blending, separating out. Dominance means the dominant character will be expressed itself, recessive character will be masked up. Independent assortment means what? That independently it is just showing which is the character of the dihybrid cross, where both the monohybrid cross, mono character will show its particular character. Now after this mutation is being studied. What is the mutation? Mutation means a sudden change in one or more gene. Particular any genetic trait is being present, any particular genetic trait is present, sudden change will occur, then it is called as mutation. Here mutation, two example is there. Sickle cell anemia. What is happening in the sickle cell anemia? Sickle cell anemia, the blood, it is a blood disease, gene disease, genetic mutation. What is happening? The particular shape of the RBC. We all know that the shape of RBC is oval. The particular shape of RBC is just becoming sickle shape due to the mutation. And in the radioactive radiation, what is happening? We all had seen that the World War II 1945 in Japan, we had just given the radioactive element. So radioactive element just mutate the particular genetic criteria of an individual. Still the particular peoples and individuals are abnormal due to that radioactive activity. So this is the particular type of mutation. So what we had studied today? We had studied today the monohybrid, dihybrid cross and its ratio. So we had just cleared about that phenotypic ratio means the morphological character, genotype means the genetic character. So we had studied monohybrid, dihybrid cross and the Mendel's three law and the mutation and its two example, and then we all had seen also today the pedigree analysis and X-linked inheritance. If there will be any problem, you all could consult me anytime. I am there to provide you, help and guide you and go through the particular chapter.